Okay, welcome everybody to this first of our online seminar series with Professor John Girkin. I'm going to introduce John very briefly and pass over to Carl for a couple of minutes to say a few words. Carl is our Director General at DIRDI, and then really I'll just leave it to John to give what I'm sure will be a fascinating webinar on biophotonics. Just a few notices from me. We have upcoming on the 25th of May, our next DIRDI dinner. That is just for DIRDI members and fellows. So if you who are part of the Institute are welcome to join, although I will say that there is limited space. So when the invitations do go out, please get back to us as quickly as you can. And then another event that's upcoming is on the 6th of June. We'll be doing our annual drone competition, and that is open to anybody to join. Anyone at Durham can join that and take part. Further details for that will be on our website, which is www.dirdi.org. I'll pass over to Carl now to say a few words.
Thank you, Daniel, and good afternoon, everybody. This gives me an incredible pleasure to introduce Professor John to you all, and many of you will already know him. But 10 or 11 years ago, Professor Paula Chadwick recommended that I speak to this incredible man. I always feel that the most eminent of our scientists are also some of the most modest, and certainly Professor John is that. And it gives me great pleasure that Daniel has created this fora for some of the United Kingdom's greatest scientists to be able to have some time so that we can see a little bit more between their modest lines of how they describe themselves. So on that note, I'd just like to say thank you, Professor John, for your support to the Durham Institute of Research, Development and Invention, your constant support to the team that is co-located with yours at the Centre for Advanced Instrumentation. And then may I just say thank you for your contribution to British science.
Thank you very much. That genuinely makes me feel quite humble after an event I attended yesterday in York Minster. So thank you very much. I will start hopefully by sharing my no. It's now asked me whether I want to update my drivers, which is the last thing I want to do. And hopefully, right. I am assuming you can now see my slideshow. I feel at the end of it, there will be some time for asking questions, but if a question occurs while you're going through, please feel free to type it into the chat. I will not answer it directly because trying to do the chat and the other bit all at the same time causes the odd complication occasionally. But if there's something that occurs that you, while I'm speaking, do put it in, you feel free to put it in the chat. Right, so biophotonics, a marriage of science. It's really just a talk here about how one goes about problem solving in an interdisciplinary manner. And that's a good start, but my slide advance didn't work. So, right, here we go. I'll do it this way. Right. We're now in business. Right, so I'm going to use biophotonics as an example, and I followed it through as a marriage. So an example of courtship going all the way through, as you'll see, leading to actually something positive coming out at the end of it. And it's really just a way of hanging the story together. I thought, oh dear, that's right. I'd give a little bit about my background. So my PhD is in atomic laser spectroscopy of atomic hydrogen. So very much not what I'm doing now. And after my PhD, I worked for 10 years in industry with a small company that formed in Cambridge area, which and some of the instruments I made there, particularly fiber manipulators, are still available in the Thor Labs catalogue. So something must have been right. And then I moved to Keeler, which is an ophthalmic company based in Windsor in the UK, and I developed the world's first ophthalmic diode laser. And if you've ever been to the optician and had a puff of air in your eye to measure the pressure in your eye, I'm sorry, I have a level of guilt there because I developed the puff tonometer as well. So And my interest now is in developing novel methods to solve, in particular, biological challenges, but they're very often broader than that. So, for example, not something I'm going to talk about here, it's how you use essentially advanced optical microscopy and adaptive optics to image what happens in a washing machine for Procter & Gamble for their low temperature washing. So that's, I've built washing machines under microscopes. But you'll see what I really want to get over today is just the overall and general approach to the problem, and particular how you can transfer technology from one field to another. So what is multidisciplinarity? What is a true multidisciplinary approach? I think it's an integrated approach. So the start is you table a significant question. Now, that could be a research question or a development question with industry, or it could be a more academic question. And you might start then by explaining, what do you think you need? And this comes along. So, for example, somebody might come to me and say, could you build a low-cost confocal microscope? And the answer is, well, yes, I could. It's not really that interesting. What do you actually want to do? What do you want to image? What do you want to measure? What's the question? And then you go, well, I could build an imaging instrument that does this. And then we can go a bit further and we'll go and speak to some chemists who might be able to add in some novel compounds to enhance the imaging. And we'll speak to some mathematicians or engineers who may be able then to develop a model of what's going on. So that's, think broadly what you need. Crucially, have the right environment where there's no dumb questions. I could give multiple examples of this, but you need to develop confidence in the group that, it's not a dumb question, because if you don't understand what somebody's saying, get them to explain it in the language that you understand. Be open, listen to alternative strategies. Don't go in, well, I want to do this multidisciplinary project and this is where we're going to do it. No, if it's got some chemists involved, listen to the way they want to do it, because it may do. And if you're looking in the end in an integrated project that might go beyond multiple institutions, multiple departments in a large company, choose the politically most suitable person to run the project, or the person with the right set of skills there. And if I'm doing this in an academic environment, but actually it goes into UKRI in general and Innovate, there's lots of benefit from larger funding opportunities because it can do more. And clearly in this case, I've said if I'm an academic, I can get higher impact papers. The other side of that is you actually get products that work very often much better and are much more profitable as they go through. So there's good reasons for doing this in all areas. So this is not a philosophy question, what is marriage? And I could ask this and could get horrible lot of questions, which will get us into trouble. But in my case here, I'm saying it's the biology bit for biophotonics and the photonics bit, which is the way of harnessing and controlling light. And you put the two bits together and it's looking at the interface between biology, and that includes clinical aspects here, and photonics. And so what's the philosophical approach that a physicist or a physical scientist takes? Well, generally, you first of all observe something, then you might build a model of it, and from that model you make a prediction, and that prediction is you then perturb your model or what's then in the real world and see and observe what happens in the real world, and then you hopefully have this virtuous circle. And this might be looking at, for example, and I deliberately use the words four-dimensional cell here because you've got X, Y, and Z and time. So, and the technology and the methodology in terms of biophotonics clearly has to help the biology. And I can look at this in different scales, and I use this when I'm speaking with physicists very often to explain the vast sense of dimensions that you're going over. So if we start in the top right with stem cells, that's typically microseconds for chemical reactions and microns for spatial extent. Then they go to build tissue patterns, which might be hours or millimeters. And then you go through to organs, which is then weeks and centimeters. And then you can go all the way up to organisms. So we're then talking, 70 plus years, meters for spatial extent, or if you want to take it to trees, thousands of years and hundreds of meters. And when you put all these together, this is not looking in 3 dimensions, but just in 1 spatial dimension and 1 temporal dimension, you end up with something which is 1 times 10 to the 27, which is 1 with 27 noughts after it, which is the sort of scale that people look at in terms of cosmology. In fact, it's bigger than many things it looked at in cosmology. So these lead to really big problems. So that's the philosophical approach. What happens at the first meeting? Well, you understand in this case what the life science researcher wants. I've got my optics toolbox, so I know my toolbox. And then I can look to go, which are the right tools to choose? And is there something available from another field? And then crucially, choose the right tool for the right jobs. And I apologize for the analogy I'm going to use. You could cut your toenails with a hedge trimmer, but for clearly obvious reasons, this is not the right tool for that job. So if you want to do some imaging that's really simple, works very well, a webcam microscope for £30 might be exactly what you need. On the other hand, if you want to, as we'll see in a little bit later, to go into some real detail with high resolution, getting some very interesting time information, yes, that's when you bring out the big guns, if you like, that might cost £500,000, £750,000. So choose the right tool. And then the other bit is, how do you go about the courtship? So I thought I'd use this example. Doctors have always used their eyes. So, you know, when you go to the doctor, apologies if there are any clinicians, because I'm about to insult them in the next couple of pages. You go there, and if you go in carrying your leg, the doctor might go, oh, he's probably got a problem with his leg there because it's fallen off. I've used very silly example, but they will look at, they use their eyes. You walk in, is the patient pale? Are they yellow? Does that indicate jaundice? The other thing is, well-known physicists have often dabbled in medicine. So Archimedes, who's really a polymath, dabbled in medicine. So he thought about the eye. And he thought that stuff came out of the eye and then hit something and got bounced back in, and lo and behold, that's how your eye works. So clearly physicists don't always get it right. The interesting bit here is that many physicists and engineers will know about Helmholtz and magnetism and a whole load of things he did. He actually has the patent for the world's first ophthalmoscope that he did in about 1860. So he was dabbling here. This is for imaging the back of the eye. There are other people that have gone to the eye. So Newton, when he was away from Cambridge in Norfolk during the Black Death at one point, did some experiments on the eye. So he took a leather bodkin, which is a leather needle, a needle for sewing leather, and pushed it round the back of his eye. This is not one to do at home, please. And wobbled the back of his eye and reported how it changed what he was looking at. There's other examples of Thomas Young also working on the eye and had a pin at the back of the retina to work out whether the eye changed its length during focusing. So people have done them, as I said, perhaps not ethically anymore. I mentioned Helmholtz. There's actually, apart from Newton, who was the Lackensian professor of mathematics that Stephen Hawking also held, there was another professor, and this of you may know is someone called Charles Babbage, and this is his difference engine that's in the Science Museum in London. He also, at the same time as Helmholtz, invented the ophthalmoscope. And the Helmholtz one uses a partially silvered mirror so that the light will shine off it. You can look through it and the light gets shone into the eye, comes back by you looking through it, and you can see the retina. In the case of Babbage, he put some little holes in there so the light gets reflected off the mirror, and then he looked in the mirror. You look through the holes in the mirror and you can see the eye. And modern ophthalmoscopes, handheld ones, still work on these same 2 principles today. The other part with courtship is to develop understanding. And again, apologies for what I'm about to say for any clinicians. You need to get to know the other person's language. And this applies not just to clinicians, but everywhere. They know a great deal, but it's about a different subject. And in particular, when working with life science data, if you're an engineer or a physicist, not all data is going to be perfect. And there's some examples that I could give later on, but just shows, you know, if we do an experiment in physics, we get a straight line, we expect all the points to lie in it and a correlation coefficient of about 3 nines, and we go, yeah, it's probably linear. If it's 0.8 in the life sciences, you go, it's pretty good. The other thing is, particularly working with clinicians, though I also now accept this may apply to physicists, you have to remember the AK ratio, and that's that arrogance is inversely proportional to knowledge. Right, so the proposal, how do we formulate the challenge? So you have a life science challenge plus the photonics, optoelectronics, and that equals your solution or your product. And there are 2 routes here. You may create new technology, or you may steal some technology. And essentially here, I've got examples of both as we go through this. The other thing is that when you marry, you normally leave home, which means you may go to a facility. So you may be in a laboratory or a setting which is not your normal physics home. So this was a center that I've had built previously, and clearly it's focused on the microscopy, and they are set up as biology labs, though in this case, there was still a bit of physics next door on the table. It's actually the same picture. We've just swung the camera a bit round so you could see that there was some physics in there. So the life scientists had to learn about the physics that's in here. So there's a learning. And the other one is, yes, try and keep the house tidy. Right. So then we come along. This is a terrible pun, and I've taken this analogy far, far too far. But one can look at it, and we'll look at some natural conceptions, some novel ideas. Once dental diagnosis is a little bit of an exaggeration, but you'll see, and also where it comes from DNA sequencing. And then the other one is how you adopt technology, particularly for imaging in the lung. So we'll now talk about some real things. So this is particularly, this took place actually some time ago now, when my eldest son had some orthodontic treatment. And orthodontists are brilliant at creating dental disease. They are magic at it, because it's very difficult to clean around all the bridge work that's set on. And it's known that in those bacterial plaques that form, there are fluorophores that fluoresce. They're generally excited in the blue and the ultraviolet, so 350, 490 nanometers, and they emit in the red. So I was aware of this. So was there something I could do for my son to help him keep his teeth clean, literally? And this was the time when blue LEDs and the ultraviolet LEDs were relatively novel. And they were only available for in the last 5 years when this idea started running. At the time, they were cheap. I mean, now they're pence each, but they were about £3 each, which was good value. They're the core, basically, of all white LED lighting. And they use low power, so 4.5 volts. Oh, that's 3 small batteries. They're small in size, and we can use plastic filters to remove the light. So what we came up with was, looking at the bottom of the screen there, we had 3 1.5 volt batteries, which gave us 4.5 volts. They went on to an LED, which was a UV 405 violet LED, which you could shine into the mouth. And while you were looking in the bathroom mirror, you wore a pair of glasses which had orange filters in them, essentially just orange glasses, which blocked the 405 light, but it lets through the red light that's fluorescing. And this enabled you to then see, or my son to then see, if there was, after he cleaned his teeth, whether there was any plaque left, and he could go back with his toothbrush and clean it again. These are some images. I should add, this is not my son when he was about 8 years old. He didn't have a moustache and all the bits. This was a dentist from Dundee that I worked with at the time. And in this case, he came into the lab and he hadn't cleaned his teeth. He'd eaten a couple of bananas in the morning and hadn't cleaned his teeth. Bananas are quite good and for growing plaque, but it comes off very easily in the case of bananas. And you can see here, I imaged it. He held the device in his hand and I imaged him through the orange piece of plastic, and you can see between the teeth there's this red area that's glowing, and you can see it in the bottom two pictures as well, and that was the area where there was some plaque developing on his teeth, and we then used some floss to clean it, and you can see that on the piece of floss you can see the fluorescence clearing very well indeed. So this was an example of taking some technology, you're aware of the bits, putting the bits together and coming up with a solution. The bit that's slightly interesting here is that this we then went to a company and said, oh, this would be good. We'd looked into it, but unfortunately it was basically not possible to patent this for several reasons. And the company, we thought you could build this into the back end of a toothbrush. It really wouldn't be very expensive, but they didn't take, two companies we approached didn't take it on because they said, well, it would be too easy for somebody to copy and various other reasons, which I think is disappointing, but it's I understand slightly where they were coming from. I still think they missed out on an opportunity. So I'm now going to move on to something different. Without biophotonics, without the photonics aspects, none of the DNA sequencing that was being done. So, you know, this time 3 years ago, everyone was talking about rapid sequencing for COVID. None of this would have been possible without the biophotonics. Yes, there's some chemistry, but there's some important bits. So the structure of DNA was Watson, Crick, and Rosalind Franklin, and the others that were involved with it. That was known. But it was then Frederick Sanger, who was English, sorry, I should say British. I can't remember exactly where he was born in the UK, who won 2 Nobel Prizes for chemistry. And he related the sequences in DNA to formation of amino acids, and then later he turned this and showed how these formed protein. And he then developed at the same time this method of sequencing, in other words, to work out what is the sequences of bases. And it was the photonics and optics that were absolutely vital to this, and the first commercial product in 1987. So how do you do this sequencing? So essentially, I'm not going to go into the details. I haven't got time in this bit here. Actually, you can look bits up. There are some wonderful videos on YouTube, and in Wikipedia is quite good. I think that's actually where I stole this image from. But the way it was done, DNA, essentially you untwist it. So you've now got a single strand, which is what's shown on the left-hand side of it. And what you can then do is at one end, it's called a 5' end, and one end ends up with a 3' end, and you chop it up into lots of little bits. And you can then have a template that it matches with, so parts of the bases that you know match. But you can also just take the primer part and onto the end of it, add that primer, you can add different fluorophores. So, for example, the red one there, if it's tyrosine, then it's red, cytosine, and then so on, and A and G glutamine. You can go down and tie on what the particular bases are. So here, the template is your original bit of DNA. We put a fluorophore on it with this primer, and it will then stick to the template. And if it sticks, lo and behold, that end will then light up red, or it would be blue or green or violet. So you can put 4 different fluorophores on. And the original way this was done was on a gel, with the DNA in a gel going through, and it would slowly be advanced by capillary action through the gel. You actually drive it electrically. And looking through on this, there's the laser on the right-hand side that generated the fluorescence, and you then detected it, so you could then see which color was coming through, which is essentially it's chromatography, but we are fluorescently labeling each base individually and being able to then read off what the sequence was. Inherently, as you can imagine, this was relatively slow. And essentially, the next generation was you would take some DNA, and there are multiple variations on it, but this is in essence the core of it. And you would tether your DNA. So that's the blue sequence that I've got here. That's my bit of unknown DNA. You've chopped up the DNA into lots of bits. You stick it on a bit of glass, so it's localized. And then you have a sea of fluorophores around there. And again, there, each base is tagged to a different color fluorophore, and you arrange the chemistry such that only one base gets added at a time. So in this one, the complement to the A is T, so it goes in and sticks down, and then in my sequence C. So you'd first of all, in this case, see some green fluorescence, and then the red fluorescence from the and then the next one would be sort of violet color. It looks more brown on my screen, and then you're going to get some green and then you go through and you would do this and you would image an area using 4 cameras and filters. You add the bases one at a time and you look to see all these flashes that are taking place, and that enables you to then read these multiple strands of DNA and to do it very efficiently. This would not be possible without A, the correct excitation. In this case, it's always done with lasers because they're narrow line width, and B, huge advances in the photonics and capability of cameras. And it's also with the high-speed detectors, actually narrowband filters, and there's chemistry with it. And third generation, which is the way a lot of it's now being done, and the bottom right-hand corner of the screen is actually a handheld portable DNA sequencer. They actually now use, it's less optically based, but it's still using physics techniques, if you like. You make a nano hole, so an incredibly small hole, which is normally photofabricated, and you strip the DNA and you then push it, or you attract it through this hole, one base at a time, and there is an electrical connection across the hole, and you monitor what the electrical activity is, because each of the bases as it goes through has a slightly different electrical characteristics. So you can essentially drive the DNA through here chemically and then read off each base one at a time going through the holes. You can clearly do multiple holes, so you can read multiple strands of DNA, and then there is a computer process which then assembles them into the right order. So the technology here is, how do you form the holes in the membrane, the precision electrical measurement, and as I said, the light is actually involved sometimes to make the hole. So photonics comes in here. So this is just an example that without this multidisciplinary aspect of putting all these bits together, you would not have DNA sequencing in the way it works at the moment. So my third example is a large project that actually financially ended at the end of March. And this was a very large project funded by EPSRC that involved collaborators at Heriot-Watt, who you'll see have one aspect which do micro machining, the University of Bath, who pull optical fibers, and the University of Edinburgh, who do, in this case, novel chemistry and some novel detectors. And the whole idea of this project, which was called Proteus, was to image inside the human lung. This was pre-COVID, so it started in, I think it was 2014. And the idea was that this is for patients in intensive care. And a terrible thing to say is that somebody comes in, and an example was given by the clinician we worked with here, who was a very fit man with a relatively young family. He was in his early 40s. He went running, and he came in with spots on his lung. Now, it was quickly diagnosed that these were not cancer, and it was therefore, and he very rapidly became ill and had to go into intensive care. And essentially, the problem was he either had a bacterial infection or a fungal infection or a bacterial infection. And unfortunately, if it's a bacterial infection, you would give probably you would give antibacterial compounds such as penicillin. But nearly all antibacterial products, that are chemicals that are used, are actually growth promoters for fungal infections, and vice versa. And so therefore, they didn't really know what to treat, and using a large imaging endoscope gives you a wide field but lacks resolution, and they had no other way of detecting this. And unfortunately, after 4 days, 5 days, this patient died, and they basically didn't know what to do. So the idea was, could you have cellular, subcellular resolution imaging through a very thin optical imaging system, and you'll see what that is, and actually put next door to it something where you could introduce chemicals, and also something where you could put fluorescent beads to monitor the physiological conditions locally inside the lung. So the chemistry, it's called smart probes, were to measure things like could we label things like bacteria, neutrophils, pH? Could we measure oxygenation? Could we measure stress? And this would all be done with fluorescent sensors. The fluorophores were known, but we needed a clever chemistry motif to attach to the relevant aspects. And the optics, the photonics, we could do optical spectroscopy, so look at different wavelengths, and also look at different fluorescent lifetimes, and I'll explain what that is in a moment. So the smart probes would be injected potentially straight into the lung, which is why, if you remember, there was that little hollow core, or we could aggregate them onto microspheres and using a suitable wavelength, interrogate the microspheres which were on the end of a particular optical fiber. And this is an example of some silicon microspheres. These are for doing an oxygen sensor and a pH sensor, one of which has a fluorophore called TAMRA. The other one is a standard fluorescein-based dye, so a sort of fluoresces in the green, one in the red. And there was another larger compound at the bottom, which has a palladium compound in the center, and fluorine, very large molecule, which was actually good for the oxygen sensing. And so this was a way where we could actually now measure the physiological properties. So that's good. The optical fibers, we want 1 micron-ish resolution in the end of an optical fiber, and we couldn't just use a single fiber, so we make, or we don't, Bath University in this case, make coherent optical fibers. So essentially each fiber at the end, so if I looked and I lined up the 2 spheres, in the end we actually used a rectangular shape, but the fibers are spherical. If you line up the bottom left-hand corner at one end, it would be bottom left-hand corner of the fiber at the other end. So there's direct one-to-one bowing, so they're coherent. Every fiber in comes out at exactly, essentially the same point at the other end. And each of the fibers here, the overall thing had to be about 1.3 millimeter diameter, but we wanted between 8,000 and 30,000 single core fibers, so a few microns in diameter, with, as I said, the catheter and the sensing fiber. This is what the end of the product looked like at the end. So crucially here for the fibers, you have to pull all the fibers. So the way you make a fiber is you make a big glass rod, and then you pull it in a furnace, and then you chop it, and then you pull it again, and you pull it until it becomes very small. But in this case, you pulled it and stacked it one on top of the other. You don't want the fibers next to each other talking to each other, so that's why you'll see that they all have a slightly different. So there are 3 sizes of fibers in this one: 1.5, 1.9, and 0.22. And the overall diameter of this thing was 6,000 microns, 0.6 of a millimeter, and we end up with a field of view of about 0.5 millimeter. And we deliberately have to make the sizes different so they don't talk to each other. And so this is what the end looks like. You can see the fibers are different sizes, so that, and there will be no 2 fibers that are the same size next to each other, so we get minimize the talk of crosstalk, the different so-called modes going down the fiber talking to each other. We end up with a pixelated image inherently, so there's therefore actually some machine learning that needs to be done, Gaussian blur, which removes that. The other thing is you don't couple the light out of the fiber in exactly the same way. So we need to then remove this so that each pixel has the right level of intensity. And this gives you an idea of what the pixelated image looks like. And then on the right is actually bin counts. Again, this is what can happen, but you can manipulate the image because we know exactly which fiber is in which place, so we can reliably manipulate the image. And the other one is the resolution. So we wanted them around, the bacteria are around about 1 micron, but they're moving. And that means that as they move across the field of view from one fiber to another, the light goes down one fiber, then the next, then the next. So essentially the light from that bit will blink, and we can enhance that effect. So we can do that. We can integrate the chemistry. So this was a part. So this is where the ends of the fiber were micro machined, and we can then put those small beads in the end, and we can interrogate each one of those beads down the optical fiber. And the inhuman results here. So this is with one of these fibers with the different beads on the end. It goes in. I won't go into the optics, but essentially we have a light source at the bottom, which goes in. There's a beam splitter cube, sends the light in and out of the fiber, goes back to the spectrometer, and we can then look and image. And we can therefore look in this case to measure and see what happens. One is, this is electronically in black for so-called optrode, which is the way it was done before. And we can then, this is, sorry, the optrode is the way we do it. The other way is we put a commercial probe in and we can test it and look, and we can clearly see that as we perfuse it, as we change the different levels of oxygen that's in there, we can see what's going on. We actually then also did this for imaging, where the first one was again a camera. So again, we have some LEDs on just that go into the microscope objective, that goes into the fiber. We image down the fiber, comes back. The image at the bottom is of a test tar bit. Again, it's pixelated, but again, we can look with two different wavelengths of excitation. We can use a color camera, and we can then pick out, we excite with one wavelength an image, another wavelength an image, and build up images now that are spectroscopically. And this is being used relatively routinely now in man. And these are some images that were taken. The person on the right in the blue and the green overalls are the clinicians, and the person in the white is the postdoc who actually built this instrument on our design. And you can look, there's a problem, you can see the background of the lung, the fibers, there's no label, and then we can look with the bacteria now labeled in red. You can see these red dots appearing. We have a problem, you can see here, there's a lot of green background. That's actually elastin that is in this damaged lung. And we need to try and get rid of that and separate it out. Luckily, it's in the green, so we can separate it from the red fluorophores. But in a moment, we'll come back to how we have a better way of separating it. So this just shows by putting it together, we can do this imaging. The other part is using some CMOS technology with things called SPADs. These are originally the driver for cameras in mobile phones, and one can purchase space on a wafer to get your own design done. It's part of what's called silicon photonics. And they have the ability to make a very, very fast detector down at about 10 to the minus 12 of a second. So this is physically what happens. You have a small area at the top in yellow where a photon goes in, and that causes an electron to be produced. But this is in a very, very small device, a few microns thick. You put a few volts across it. That means the electric field is very, very high. So that electron that's been produced from the photon gets accelerated. It hits a part of the other material that gives you 2 electrons. Those 2 electrons gives you 4. So you get an exponential breakdown. So for every photon that goes in, you can get a relatively large electrical signal. And this enables, it happens very, very fast. And what we can then do is use this fast detector. We put a light pulse in that starts a clock, and then the photon stops a clock. So in period 1, we didn't get a photon. In the second period, oh, we've got a photon this time, so the clock has stopped. Period 3, we don't get anything. Period 4, we get a later one. And essentially, we can then build up the timing of each of these photons, and you end up with the histogram at the bottom, which is the number of photons against time at the bottom. And you can then plot this out, and you get an exponential decay. And most fluorophores that we're talking about decay in a few milliseconds, and this means a few nanoseconds, and this means that we can then see what's happening in real time and use this fluorescence lifetime to see what's going on. We need high-speed electronics to do all this, But actually, using CMOS, we can actually build the electronics behind the light sensor chip, which is what they do in mobile phones. So we've got that. And this is a linear array, which has 512 pixels, 50 picoseconds. So that's 50 times 10 to the minus 12 temporal resolution. And we can either count each photon in time, which is the TC-SPC, time correlated single photon counting, or we can put them in time bins. And this enables us to do imaging at 15 frames a second and to get this information on the fluorescence lifetime, how the fluorophore decays with time. And its decay with time is very closely related to its environment. And this is then the scanning system that we built with this. So LS1 is the laser. We have a second laser that's the option of going in there. It goes into G1, which is a scanning galvo mirror, which moves it in X, off a curved mirror onto another one, relayed from another curved mirror onto the second scanning galvo, which now scans it in Y. And then we can re-image that and go down a fiber. And all of those, they're normally done with lenses. We designed this to use reflective optics because reflective optics, as long as they have the right coating on or are made of the right material, respond in the same way to all wavelengths, whereas glasses have what's called chromatic aberration. They focus light at slightly different depths. So we can remove that, so we can make something that's entirely achromatic. And this is what it looks like on a bench. And as you can see, we probably couldn't put that in a hospital. The board is on the left-hand side. The scanning is in the middle, and the light delivery system is on the right. So we then took this, and how do we engineer it? That's a bit of detail of how we launched the light into the system with the scanning mirrors. And it's got to be robust. It's got to be simple enough even for a clinician to use. We have to keep all the beams aligned, keep them enclosed, and the detectors. And the solution here was to take a block of aluminium and machine all the items from a solid block. That gives you good heat sinking to remove any heat. It's robust, it's lightweight, and we have fiber connectors going in and out everywhere. So it was designed and built, and essentially you end up with the scanning optics going into our detector and then off to the electronics. And this is what it looked like. So this again it was designed by ourselves. It's all machined from a solid block of aluminium, and then you make it with a long series cutter, machine all the bits out. You have to make a few extra bits for the moving mirror plates that are going to move, but the whole thing is really very, very stable. And top left is what it looks like with the computer and the spectrometer part, which is the black box next door. We can put all this in a clinically approved trolley and wheel it around in and out, and we do all sort of tests. We drop it and we bump it and it stays aligned. And this is the spectrometer. So we have this so-called SPAD array. The light goes in, we collimate it, it goes through a diffraction grating, splits it onto our 512 detectors, and we get roughly 1 nanometer resolution with fluorescence lifetime capability for each nanometer. Generally, we grouped the buckets together, but you can make that grouping whatever we like. And there's another technique that we also used with this called Raman spectroscopy, which is just the way the light changes wavelength due to the vibrations of the molecules. Normally, that is an effect very much smaller than fluorescence, but we can separate it out using our time gating. And this is a sort of an image. This is not human lung. This is a plant. And we can set the spectral range, the spectral resolution. This is intensity in 2 spectral bands given false color. And this is the lifetime now. So this is how long a fluorophore was fluorescing. So in the image on the left, it's from 2.7 roughly nanoseconds down to 0. The one on the right is longer wavelength, longer time bands at a different wavelength as well. So we've got both spectral and lifetime measurements. And you can see even for the same fluorophore, we're getting different lifetimes, which is telling us something about the local environment. And you can then plot them as histograms and various ways of processing the data. What we then did is we then took this, and this is the way of presenting the data. So if you like, the bit in the middle is the full spectrum, and then for each of those spectrums we can take time slices, so the images that you see are color coded for lifetime. So we have both the spectral and the lifetime information in the same data set. There are issues with processing the data, but this is actually from one of the papers. This was ex vivo lung tissue, and this is an in vivo image, which I hope will play. So this is an image inside the lung. Now, again, what you're seeing there is the elastin, which has a different fluorescence lifetime, so we can actually separate this elastin out. But this is, as I said, an intensity. This is not in a healthy patient. And the next image is, I will move on to the next image. Thank you. So I have to say there's one part of this which we were very lucky, which is a horrible way of phrasing it, but this instrument was all ready to go when COVID struck. It wasn't yet approved for use in humans, though we'd given them all the paperwork, and that normally then takes about 3 to 4 months to go through. In this case, it got rushed through in under a month, and we took the first ever images of what was happening at a microscopic level in vivo in a living human COVID patient. This is one that was taken up in Edinburgh, and we labeled the macrophages, which are some, and the neutrophils. The neutrophils are supposed to be going in there, and it's what many of the people died from. It's the body's overreaction. The neutrophils are actually the good guys. They're trying to eat up the disease, but they basically over proliferate. You get too many of them. And the green is the autofluorescence within the lung of the elastin. This was also part of a drug clinical trial because we could locally introduce the drugs down the catheter that was there. And actually, this was involved in an early drug trial to select which drugs, because we could introduce them locally, potentially flush them out instantly if they caused a problem, were just used locally in the lung. And dexamethasone, which was one of the drugs that was then used normally in the treatment of the patient, was first looked at under this method. So I hope I've given you an idea of how you put all these bits together, ask the right questions, do the right things, and admit there is no such thing as a stupid question. Listen to the answers, work together. And I have to acknowledge here, Chris Longbottom was the person with the dentistry, and there is a huge list of collaborators. The Proteus project has 50+ people involved in it. I've just chosen Mark Bradley is the person, the chemist that leads the overall project. Robert Henderson is the person that designed the SPAD arrays that were in there. And Gareth Williams was my postdoc who built the optical system that was in there and drove the thing forward. He now has his own fellowship. So with that, I'd like to thank you very much for your attention. I have no idea whether there are any questions or present, but I will stop sharing my screen now, which if I can find the way of doing that, if I remember within Teams, which is down here. Right. There are some questions in the chat. Oh, no, they're not. It works. Right, they were all questions. I can't see anything. Right. In which case, has anyone? Yes, I stick your hand up, but I'll let Carl go first because he's physically in the middle of the screen and has stuck his hand up.
Professor John, thank you so much for that, and I warmly congratulate you on just. I happen to know that that's just a brief summary of your major contributions. I wanted to ask a question about the Centre for Advanced Instrumentation. So, at what point in your career did you think that that was necessary, and how long did it take you to establish it?
I think so. I didn't establish the Centre for Advanced Instrumentation, but I'll take a step back and talk in a slightly different way. So I was originally brought from Durham. I was one of the founders of the Institute of Photonics at Strathclyde, which then became the first Fraunhofer in the UK. And I was brought here to form and get going the Biophysical Sciences Institute, which is how we do some of this. But I was assigned within physics to the Centre for Advanced Instrumentation, which at that point had been going about 10 years. But having said that, it was originally set up, it was originally called the Centre for Astronomical Instrumentation. And of course, I don't do astronomy, but I use many of the techniques that are used. So it was changed to become the advanced, or part of the reason to advanced instrumentation, when we were often taking astronomy techniques that were put on big telescopes and putting them down a microscope. And then it broadened out and then said, well, actually, the techniques that were being used on telescopes for imaging the stars, and one of the areas we're now working in, are very important areas for driving forward optical communications via satellites. One of the big problems with satellites is getting the data off them, because radio is too slow. So you can do it optically, but that means you've got to go through the atmosphere. So you have to remove the turbulence in the atmosphere that affects the optics. So it was formed in this way that would enable multidisciplinary and particularly large projects to take place. So we have a team, a bit unusually for an academic environment, of engineers who are working all the time on these projects. Many of them have PhDs, but they're not so much doing research projects as developing the instrumentation. So it has a slightly different, they're rewarded in a different way. We're now actually looking to actually take this, in some bits of it, in a slightly more commercial direction as well, because we do build instruments. We built instruments for Airbus, for example, because of the techniques that we've got. So we're looking to broaden it. So it was this way of how do we get the optical engineers, the mechanical engineers, and also then to collaborate with people from other departments and other institutions together.
So 2 questions then, thank you. When it was the Centre for Astronomical Instrumentation, would that have been something that Professor Paula Chadwick had been involved with?
No. Paula is part of the Centre for Advanced Instrumentation, but when it was originally set up, it was mainly set up by someone called Ray Sharples, who built spectroscopic instruments, and it came from that. Though, yes, when it was formed, Paula then became part of it. She wasn't one of the original driving forces, but yes, she does different things. So she's looking for high energy gamma radiation, which you actually detect generally using optical detectors very often. So yes, she's part of this because her instruments involve optical instrumentation, and therefore we have the skills and also the image or the data processing and high speed electronics that's needed. So again, that's she's looking at things very different from conventional things. Like we've got one of our instruments is on the James Webb telescope. Her instruments are very different from that, but at the core, they use the same technology or the same methodology.
I remember when I was getting to know Professor Paula and then Dr. Douglas Halliday, and now Professor Douglas.
Yep.
I remember being very proud of myself once when I was actually able to describe her to somebody as a ground-based gamma ray astronomer.
That's right.
And it had taken me weeks to be able to be fluent in that. Secondly, when you mention engineers at the Centre for Advanced Instrumentation, would you agree with a little thought that I've had over the last year or so, that the government is describing science and technology better than it has in previous years, and acquainting a greater part of the country with the importance of that? But I still think it's missing the binding link of engineering in the middle of those 2 words.
No, so in terms of funding, I sit with the Engineering and Physical Research Council. So in that sense, they do view it all as the same thing. No, I completely agree. And part of it is what I'm going to call for the moment a UK problem. So there is still a perception, I think, within the UK that the engineer is the person at the garage that fixes the brakes on your car. And,
Right.
A horrible cliche that gets their hands dirty with oil. Yeah, some of our people do get their hands dirty with oil because the instruments need to be oiled. But whereas if you went in, and you look at the heads of many companies in Germany, for example, they're often headed up by engineers. with PhDs in engineering, whereas that is less likely to happen. And the technical directors in companies in Germany, I'm using particularly Germany, yeah, they're often technical, they're qualified engineers. That happens less within the UK. So there is still this perception within the UK that the word engineering means, inverted commas, the person with the oily rag. We may have got beyond it being the man with the oily rag, so we're getting better, but it's still this perception. And I completely agree. And I have, if you ask me a different question, and I don't know the answer, is when does engineering stop and physics start, or physics stop and engineering start? I'm not quite sure.
Or you could say,
I have some thoughts, which is, you know, but it's a continuum.
Or you could say that engineering is the enabler for much of science and technology, but either way, the sequence in its description would be better, it seems to me, as science, engineering, and technology. And then you really are attracting that German aspect, which we need so much in the United Kingdom. Look, I don't want to monopolize your time, but I'm very grateful to you for your many contributions to British science and your support to the Institute. But Daniel, if you're there, perhaps I should turn this over to you.
Thank you very much, Carl. I think we're just about done with time. So all I will say is just thank you very much, John, for that wonderful seminar. I had absolutely no idea that you were involved with those very first images of the human lung during COVID. But on the whole, fantastic webinar. And if anybody does have any questions, feel free to email me and I'll direct them to John. But thank you everybody for joining, and thank you, John and Carl.
Thank you. And I hope people enjoyed it. And as I once said, I hope I had this comment once, apologies, I think Carl's heard me say this and some others may have done. I had a comment when I was teaching second year quantum mechanics and atomic physics, which said, I hate this lecturer, not a good start. He makes me think. Now, I took that as a positive. And if there's one bit that you take away from this, I hope you don't hate me. I hope that it's made you think. And if I've done that, that's my aim for this thing. Thank you very much.
If I could step in, you have. And the other thing that I'd say is that this really should be a platform for senior British scientists to be able to describe what really brought them into science in the beginning. And I thank you so much for showing us aspects of that today,
That's
Professor.
The space race in my case. There you go.
There you go.
That's why I do it.
All right. So okay. God bless you. Thanks
very much. Okay. Bye.
Thank you, everybody.
Thank you.
Bye-bye.